Good afternoon, everyone. My name is Song Jaejin. I'm professor of biomedical science. What I'm going to present today is about the mesenchymal stem cells capable of enhancing tumor targeting and mass viral production. To help you better understand, I brought this image. I hope it will improve your understanding. So this is the schematic diagram for the research that I'm doing. This is the form of the viruses, and developing the cancer treatment is what I've been doing, and this is the main research that I've been involved in for the last decade. And this cancer treatment, in order to help the patient treatment, there is a one big major hurdle that needs to be overcome. And that hurdle is this: the delivery and targeting. These are the two major hurdles that needs to be overcome when we develop the cancer treatment. That you see the virus-based cancer treatment. For this treatment, we need the very successful and efficient carrier for drug delivery and targeting. And for that, I've used the mesenchymal stem cell. And mesenchymal stem cell was well known as effective for treating the cancer. But there were some limitations involved as well. So in our research, we studied the mesenchymal stem cell. We modified this stem cell. So oncolytic virus that are poorly delivered to tumors upon systematic administration. And it was developed back in 2000, but now we're in 2021, but not much improvement has been made because of this issue with systemic administration. Most researchers, including myself, were studying efficacy, safety of the drug candidates. So the majority of the studies were focused on efficacy and safety. So efficacy and safety has been improved over the last decade, but to have critical relevance, the delivery is the most also the equally important factor, but these needs to be addressed. So tumor targeting is the issue that needs to be addressed. So in terms of efficacy and safety, a lot of improvement has been made, but this oncolytic virus drug candidates have not been really made into the drugs because of this delivery issue. So how we address the delivery issue will address this issue. Let me take another example, and this was also released early 2000. That the IV injection is the main form of delivery for this oncolytic treatment. That you see that from this figure that you might not be able to guess that how much is being delivered to the tumor. But oncolytic viruses are circulated by the liver or spleen, and 0.001% of the administered virions were found to reach the Hind-Frank tumors. So let me summarize the three major hurdles in systemic treatment of oncolytic adenovirus. So in the case of systematic treatment, that these are the major hurdles. First, the sequestration in the liver, And second, inefficient delivery to the tumor site. So to the tumor site, only a little amount is being delivered because of the delivery issue. And the third, the rapid elimination as a result of immune clearance. That, the Steve Jobs passed away in 2011, and the soccer player Yu Sang-chul also died of the prostate cancer. And this player, Yu Sang-chul, soccer player Yu Sang-chul, died of pancreatic cancer. And that you see the case of this player, he has the characteristics, the major characteristics of the death of pancreatic cancer. So he was diagnosed at the later stage, the fourth stage, and he got the anti-cancer treatment, and he died after metastasis. So there are unmet medical needs for cancer treatment. So if the diagnosis is made at the later stage, that it might be better if we can have the diagnosis in the earlier stage. And there might be a lot of side effects coming from injecting these many different types of the anti-cancer treatments. So if we can make the treatment more effective, that will really address this issue. And this patient died after metastasis. So even if the patient has metastasis, it will be better if there can be the treatment that could be applied for this stage. I brought this photo as well. You might be wondering why I brought this photo. As I've mentioned, the oncolytic virus treatment, the issue with this treatment can be likened to the case of Trojan horse. So that's why I brought this photo. As a carrier, the mesenchymal stem cell is applied, and mesenchymal stem cell is just like the Trojan horse. You see in the castle of the enemy that the Trojan horse was moved into the castle and the fortress of the enemy, and at night the many soldiers were coming out of this Trojan horse. That MSC is working as the Trojan horse. But the existing MSC does not have the enough potential to defeat the soldiers, so we need the stronger soldiers. So we need to modify and enhance MSC, and that was the major purpose of our research. And as you can see, so within the fortress of the enemy, the Trojan horse was there, and it was delivered to there, and with the help of modified MSC. And within the Trojan horse that you see, a lot of soldiers were hidden there. Like dozens of soldiers were hidden there. And we might be able to increase this number of soldiers, but not just the number. We might be able to increase the arms and the capacity of the soldiers. So just like the modified Trojan horse, that we thought of applying the modified MSC. So that was the main purpose of our research. And I'm not sure it might be easy for you to understand, but let me take the analogy. So if you see the rocket, the upper part is the engine. Without addressing this issue with the engine, the majority of the studies were focused on the oncolytic viruses themselves. And some other genes were injected into the drug candidates. and the treatment efficacy was going to be improved in vivo. So you see the head part. This is the loaded genes. And by having a different type of combination of genes, that we might be able to have the better treatment effects. So the combinations effects and were confirmed and validated, that you see that these genes were applied, and how we can have the both tumor versatility and specificity, and what kind of genes might be able to be applied. So these were some of the things that were studied in our research. So loaded genes combination, that we also obtained the patents. And for the body, the virus structure, we also made a contribution for this part as well. So the virus replication and amplification. So we also have the findings from our study as well. But with this too, the head and the body part, we confirmed that in vitro that there are some efficacy, but in vivo, if we will be able to be applied this to in vivo, that we need the right delivery. So without having the good delivery, that these two parts might not function really well. So since 2 years ago that we've conducted the study on delivery, MSC delivery, and just a while ago, together with the PCT, that we have obtained the patent on this. As you see that there are from numbers from 1 to 7, and with these factors, the delivery issue was addressed. So method that we use is the oncolytic virus and mesenchymal stem cell. It's a combination of these two, and it's a platform technology, including a lot of genes and the virus replication features. And the pipeline is that it's a nodulation type of this, and this is the findings of our study. With the simple combination of genes that we've conducted the study, and as you can see, the results are like that. With two genes are combined, and for many genotypes, that regardless of the aggressiveness, that you can see the effect across the various spectrums. And like I've shown earlier, this is showing the targeting, tumor targeting with versatility. And with this, I think we have made a really significant upgrade. Choi Soo-jin, the graduate student, was leading and was involved in this study, and we were able to be confirmed that this can be replicated in many different patients. IVIS, and that you see the difference between the treatment method. So the tumor was injected on the shoulder part, and for that part, that we may use the fluorescent to make it into the different color. So the two different MSC was applied. One is controlled and the other one is the candidate that we have, the modified MSC. That you see that in many different tumors, that you see the versatilities across the many different types of genotypes and many different types of cells. Yes, also there is a room for further study, but still that you can see that it's working across the many different tumor type. So it's quite versatile. With these findings that you can see within a really short period of time, as opposed to the controlled MSC, that this therapy is being delivered to the tumor on the shoulder part. An intensity that it's difficult to objectify these figures, but depending on the tumor type, around 100 or even 1000 might be able to be formed there in the tumor part. And it's different from the existing MSCs. So the carrier cell that we have is an improved version of modified MSC. So there is another concern. Yes, the fast delivery to the tumor is possible, but how about to other organs? And this is also the results of our study. To objectify the results of our study, so we've commissioned the research to the research institute that you see tumor, liver, spleen, lung, that you see the results for each body organs, the maximized figures that you see the value is like that. And in other body parts, if you look at the intensity, it's not no higher than 100. So it's a quite different level. So in most cases, I don't think that this term, in most cases, is relevant. So you can see that this is going directly to the tumor site without having any inhibition. And this can be also similar to the Trojan horse that I've shown earlier. So from the Trojan horse, the soldiers need to come out. But when? The soldiers cannot come out during the day because soldiers are there stations within the castle. So when the soldiers come out, it's really important. It's also important for this virus as well. After it's being delivered to the tumor site, without having the right regulatory system, this virus might come out at the wrong time. So after it's being delivered to the tumor site, the virus needs to be regulated really well with the right technology. So we've applied this system. Doxycycline, so there is some doxycycline and no doxycycline. So with the little amount of doxycycline, that you see the difference with the absence of doxycycline, that you see that it's switching on right away. So from virus, that specific the virus expression can be made right away, and it's working quite well. So this tells us in vivo, its doxycycline is observed. So when the little amount of doxycycline is fed, that induction can be made really within a short period of time with this regulatory system. And these are the findings of our study. One data is missing. And this is the result of in vivo study. So oncolytic virus is injected, and the results are like that, as you can see on the slide. So after injected in 7 days on the tumor site, so it's even if it's injected from the tail site, that you can see that it's being delivered to the tumor site on the shoulder. As you saw from the previous slide, for most sections, the virus proteins were detected, and the results are illustrated here in the graph. So when only NSC is injected and when the NSC is injected with the method that we've developed, the tumor targeting is done better, And with the virus that has been released, really elicit and creates a tumor aggression. And this is the result of the tumor site after 30 days of OV MSC injection. So one mice were selected from each group. So it was delivered to the tumor site that you see that it's working right at the tumor site. And this is the final result. And there is the one important factor that needs to be included, that tumorigenicity needs to be ruled out. There are some factors that is working for this ruling out of tumor genericity, but I cannot mention all the details because of the time constraints. There are some genetic mechanism that we have validated, and we're going to share that as well. So even that this tumor regenicity over time, that you see that we can even regulate the tumor genericity with this modified MSCs. So with the technology that we have obtained, the patent that we have addressed these most known hurdles of virus and cell cancer gene therapy. So anti-cancer platform technology that we established is a new type of cancer gene therapy. And finally, this is the final diagram that I would like to share with you. So tumor targeting is done really well. And when we combine the other elements that can help this delivery and tumor targeting, then this will really be capable of cancer diagnosis as well, and as well as the treatment in the near future. Thank you very much.